Well, welcome everybody. I'm going to get started with a very brief introduction about SGP itself. So for those of you who are new, and I see a couple new faces, we are an open community-oriented research consortium that seeks to address questions of environmental evolution across Earth history through statistical analysis of the sedimentary geochemical record. And if you're interested in learning more or getting in touch with us to join, please check out our informational website or our search website there. We now have geochemical data in our database for more than 110,000 samples and approaching 4 million analytical results. And this is all due to the hard work of more than 160 geoscientists and data scientist collaborators around the world. And if you're not part of this group, we would love to have you join. The talk today is part of our SGP Proxy Primer series. The goal, we found there were a number of people that really wanted to learn kind of the basics of the different geochemical proxies that we study in the group. And so the goal is to accessibly explain the basics of a geochemical proxy or method to a broad audience. We then try to curate these lectures on the SGP YouTube channel to create a Proxy Primer video library that people can access. And the other goal is to provide an open space for interaction with experts on each proxy. And so at the conclusion of this, we will be having an open Q&A that will not be recorded, so people can ask all the questions that they've ever wanted to know about cerium anomalies and rare earth element geochemistry and carbonates. As I mentioned previously, this Q&A will be led by Dr. Kim Lau at Penn State University, who is also a carbonate geochemistry expert, and excited to see that discussion. So for today, for the Proxy Primer, we're delighted to have Dr. Rosalie Tostevin from the University of Cape Town. She did her PhD at University College London before moving on to a postdoc at the University of Otago and the University of Oxford. She then started her faculty position at the University of Cape Town in 2019, where she has gained a reputation as a world expert in carbonate geochemistry and Earth history. And so with that, we are delighted to learn more about cerium anomalies.
Okay, thank you, Eric. I'll just share my screen. Okay, hopefully you can see that okay.
Yep.
Yep, cool. Yeah, thank you. I've been working on rare earth elements for some time now. I became interested in them in my PhD in carbonate rocks, and more recently I've been looking at iron silicates. So I'm going to begin by going through just the basics of the proxy from scratch, and then a few case studies from the modern environment, and then a few case studies from deep time. I'm going to talk about cerium anomalies, but also the rare earth element patterns as a whole, and I don't think we can really decouple the two. You have to understand both and use them together in tandem. So the rare earth elements are this suite of elements that sit beneath the rest of the periodic table, called the lanthanide series, and we often also include lanthanum and yttrium alongside the rare earth elements. They're present in both igneous and sedimentary settings, but I'll be focused on sedimentary settings today. And they're not actually that rare, and they're not actually earth elements. So the name doesn't make a huge amount of sense. They're more like transition metals. And on average, you know, they have a reasonable abundance in Earth's crust, but they're very dispersed. So as we move across the rare earth element series, we normally order them by atomic number. You know, the electrons, obviously each element has one extra electron and one extra proton, and those electrons are ordered in shells in a very strict order. So we fill up the S and the P and the D, and as we move across the lanthanide series and we add a new electron each time, it enters the 4f orbital, and that's one of the reasons why the rare earth elements are so unique and also so useful. So the 4f orbitals are kind of tucked away inside the atomic structure, and they don't extend far enough away from the atom to directly participate in bonding. So that means that even though we have this whole suite of elements, they have remarkably similar behavior. And then there's a couple of anomalies which result from either empty, close to empty, half-filled, or full electron shells, which imparts a little bit of extra stability on those elements within the series, and results in slightly anomalous behavior. So this is a typical rare earth element distribution pattern from modern seawater, and also just a typical rare earth element plot. So we have the rare earths listed by atomic number along the x-axis, and we subcategorize these into the light, medium, and heavy rare earths. And then on the y-axis, we plot the concentration in the sample normalized to shale, or upper continental crust, but commonly to shale. So PAAS just stands for post-Archean average shale. And I think the original reference for that was Taylor and McLennan, but this slightly modified version published by Poormand in 2012, which is the one I use for normalization. It's a very, very simple process, just dividing one number by another. And what you can see in the concentration in modern seawater, firstly, it's very low. We're looking at 10 to the minus 7 ratios down here, which is much lower than we see in the rock record. And you can see across this series that actually in general, if we ignore the anomalous elements for now, there's a very nice smooth pattern. You could almost draw a straight line through this, with ever increasing concentrations from the light towards the heavy rare earth elements. And this is a result of carbonate complexation in seawater. So carbonate complexes are slightly stronger with the heavier rare earths, which just means they're more stable, they're less likely to be removed through scavenging, and you end up with slightly higher concentrations in seawater. So we've got this nice baseline where we can define a straight line. And then against that straight line, we can start to quantify the anomalous behavior of some of the elements I mentioned earlier, like lanthanum, which has an empty 4f electron shell, and so slightly higher concentrations than we'd expect. Gadolinium also has a small positive anomaly, and slightly higher concentrations than expected. Yttrium does too. It's because it doesn't actually sit within the lanthanide series, but we include it. It's very important to include it. A lot of earlier studies didn't, but it is a really useful parameter. We can do a lot with the ratio of yttrium to its neighbor holmium. And then on top of this, we have two additional anomalies which are generated by redox changes. The first is europium, which is only generated under really strongly reducing conditions like those we may find in hydrothermal settings. And then the cerium anomaly, which is generated under more ambient seawater redox conditions. So I'll go into a bit more detail on cerium. It's kind of the star of the show within the rare earth element series. And this is just a little cartoon showing the systematics of how cerium anomalies are generated. So in oxic waters, manganese 2+ can be oxidized to manganese 4 minerals, and cerium 3, so all of the rare earth elements sit in the 3+ oxidation state except for cerium and europium. Cerium can be oxidized to 4+. And that's facilitated on the surface, catalyzed on the surface of these manganese oxide minerals. And the cerium 3 hydroxide phases are soluble, whereas the cerium 4 oxide phases are insoluble. And so they're removed preferentially from the water column under oxic conditions. And the rest of the rare earths remain in solution in the 3+ oxidation state. And then as oxygen concentrations decrease and we enter the manganese zone, so below a chemocline where redox conditions are conducive to the dissolution of manganese oxides, those solid particulates dissolve, the manganese is released, and the cerium 3 is released back into seawater. And so the cerium anomaly, which I will define mathematically later on, but is basically the concentration of cerium relative to the concentration you would expect based on that trajectory from its neighbors, is negative in oxic waters and then either positive or close to 1 in anoxic waters. So we have a redox proxy which is sensitive to intermediate conditions and operates on a relatively localized scale. So the residence time of all the rare earths is very low, and you can have variation on, residence times of as low as 1 year, which means you can have variation in between different shelf settings and within a single shelf setting with depth. So one thing that makes Cerium anomalies a relatively special proxy is that the majority of proxies that we have available to us respond to fully anoxic conditions, so the onset of either ferruginous conditions, which are sometimes termed suboxic but only occur in the complete absence of oxygen, or euxinic conditions with the buildup of sulfide, which again only occurs in the complete absence of oxygen. Whereas cerium responds to this boundary here, the onset of manganese conditions, which are slightly less reducing on the spectrum and can sometimes overlap with low levels of oxygen, or even quite substantial levels of oxygen, in some cases up to 100 micromoles, which is relative to a maximum in the modern ocean of 250 is relatively high. So that's one thing that makes cerium a pretty special proxy in my view. The other thing that I really like about it is that you don't just have the cerium anomaly, you have the entire rare earth element pattern at your disposal. And there's lots of other useful information encoded in this pattern. So one useful thing is that we see distinct rare earth element patterns in different phases. So they can be used to identify provenance and also contamination. So this means we have some kind of internal check alongside our redox proxy. We can look at the entire rare earth element pattern and say something about whether we think we've extracted a primary seawater signal, or we think we're looking at more of a hydrothermal signal or a clay signal, or an iron manganese crust, for example, which is obviously very useful when we're trying to reconstruct environments in deep time. These are just a few case studies from modern settings. So obviously modern anoxic environments tend to be quite biased towards lakes, which are commonly stratified, such as Lake Vanda in Antarctica, but we also have some marine restricted basins which are driven towards anoxic conditions, such as the Cerium Trench. So in Lake Vanda, the oxygen concentrations in blue start to decline about 66 meters. The manganese 2+ concentrations increase, so this is us passing through the chemocline and dissolving the manganese oxide minerals and producing manganese 2+. And a little bit below that, we have the onset of euxinia. So there's a very narrow band here, only about 1 meter wide, which I would call the manganese zone before we have the onset of fully anoxic conditions. And if we look at the rare earth element patterns from Lake Vanda from 66 meters, we see a big positive yttrium anomaly and a negative cerium anomaly. And 1 meter below that, we also see, some similar hallmarks of the rare earth element signature, such as positive yttrium, but our cerium anomaly is completely inverted, and it's now a positive anomaly. And what I mean by that is that if you were to draw a line through this rare earth element pattern, cerium concentrations that were measured in solution are much higher than you would anticipate if there was no additional fractionation of cerium occurring. It's a similar story in the Cerium Trench. So the oxygen concentrations decline by about 220 meters. And then we have the buildup of manganese in pink and iron in green, and eventually below about 300 meters, we have the buildup of euxinic conditions. And again, we don't have yttrium here, unfortunately, it wasn't measured. But if we look at the rare earth element patterns here at 256 meters, we have a beautiful negative cerium anomaly, and by 327 meters, we have a positive cerium anomaly. So this is just nice proof from modern environments about the close relationship between the cycling of cerium and the cycling of manganese, and that in some cases that's spatially separated from the onset of true euxinic or anoxic conditions. Obviously, when we're looking back in deep time, we have to extract rare earth elements from orthogenic minerals, and we have a number of options. So for example, some iron minerals, I'll talk about iron silicates later, but hydrothermal iron oxides if they precipitate rapidly enough, they can preserve seawater rare earth element patterns. But some work, I think by Michael Bauer, has shown that cerium can continue to accumulate on the surface after the iron oxides have been precipitated. So it may not always be a reliable record of the primary seawater cerium anomaly. And these deposits are also relatively rare. We know they're not distributed evenly through geological history. Phosphate minerals, there's some promising data showing, for example, covariation between negative cerium anomalies and other indicators of primary redox, like bioturbation. So that, there's some people that believe they're recording primary marine cerium anomalies, but they are susceptible to overprinting. They continue to scavenge rare earth elements in a non-quantitative way during diagenesis. And again, they're relatively limited in their distribution in the geological record. So I think carbonate minerals offer us the most promise because we have a relatively continuous and well-preserved archive. They're widespread, they record what's happening in shallow shelf marine environments, and there's also some good evidence that they record primary seawater signals, at least in some cases. So this is a classic paper by Greg Webb from 1999, and he measured rare earth elements in modern microbialites on the Great Barrier Reef and found that they nicely record seawater rare earth element patterns reasonably faithfully. He recorded the partition coefficient for each rare earth. He calculated that, and we can see that there's a very high partition coefficient, which is what we want to see, because remember, the concentrations in seawater are extremely low, and the higher the concentration, the easier it is for us to get good data. So there's a relatively high partition coefficient, and it's the same across the entire series. Although he did note that the partition coefficient for cerium was slightly higher than the rest of the rare earths. This isn't something I see mentioned in the literature a lot when interpreting these patterns, but I think it's actually probably a pretty important point, that the magnitude of any cerium anomaly could be reduced compared to contemporaneous seawater. We also think that rare earth elements are incorporated into the lattice, replacing calcium, and that they're relatively robust to diagenesis. So this is a massive question, obviously, for all geochemical proxies, and particularly in carbonate settings. We know that we have a seawater signal which enters the primary minerals, which might be aragonite or high magnesium calcite, and that they, over geological timescales, are going to transform into low magnesium calcite and dolomite, and that they could be exchanged with pore waters during that process. So we have a little bit of data for this. We have some older data that I'm not showing from Banner, indicating that you can still preserve primary seawater rare earth element patterns even in dolomites. And that was from relatively recent sediments where you could see the evolution of different minerals with time, and that the rare earth element pattern seemed to be really, really robust to those processes. There's more recent data from Liu in GCA 2019, looking at the Bahamas, where there's been a lot of interest recently in quantifying how geochemical proxies are impacted by diagenesis under various conditions. So they have pretty well-constrained overprints there linked to meteoric and marine burial diagenesis. And what they found is that regardless of the mineralogy, the diagenetic fluid composition, and the redox state, the rare earth element patterns, including the cerium anomaly, are similar to those of modern oxic seawater, indicating they likely record the seawater signatures of primary deposition. So there's clearly no, what I see in this data is there's no trend here as the mineralogy is changing, but there is rather a large range, and obviously some of these samples are not recording contemporaneous seawater, and some of them are. There's also a recent paper by Zhang and Shields pointing out that in this study, the fluids are largely oxic and similar to modern seawater in the pore water, so it might not be the best test because it's not actually, really pushing things. It's a relatively easy system as in terms of diagenetic alteration potential. So I talked about microbialites. A lot of work on rare earth elements in carbonate rocks focuses on inorganic or microbial precipitates like ooids or just bulk rock Precambrian carbonates and microbialites. There's not been as much work on shells. I've become quite interested in this and putting together patterns of rare earth elements from different types of biomineral. And it seems that there's some large systematic differences between, for example, like bivalves and mussels and brachiopods and coral. And these are all things that are precipitating from modern seawater, right? So this is a question of how well these biominerals are recording the signal rather than potential changes in the seawater signal over geological time. Unfortunately, all of these studies focused on individual shells and they all used variable cleaning techniques. So I'm not sure whether this is a methodological signal or an ecological signal, but potentially there's some very interesting information there that my group are starting to investigate, which is there some systematic difference in the way that some shells record rare earth elements patterns versus others, and does that tell you something about their biomineralization mechanism? Or is this just because a lot of these studies have not sufficiently cleaned the organic matter and clays away from the shells before digesting them? So we've shown with bulk rock that is really important, that you have to employ a sequential digestion technique to extract these seawater signals, because the rare earth element content of clays is so much higher than carbonates that if you even with your weak acid in a single step digestion attack a tiny amount of clay material, it can overwhelm the weighted average rare earth element signal. And this is a single step digest in a weak acid. And you can see that we have relatively high concentrations, no yttrium anomalies, no lanthanum anomalies. And then when we do a sequential digestion, which means only adding sufficient acid to digest a fraction of the sample, so 40% rather than 100%, we start to see lower rare earth element concentrations, big yttrium anomalies, and positive lanthanum anomalies, as we would expect in a seawater pattern. And this is from a paper that came out of my PhD in 2016, but it's been shown also by a group led by Zhang and also by Cao 2019, finding similar things with slight differences in the acid used, and they also recommend a filtration step in Cao, which I've now incorporated as well in my group. So I think this is pretty robust now, that it's really important to use this partial digestion technique. And I think a lot of the older data in the literature that weren't using this technique potentially have suffered from significant clay contamination, which would have obscured any cerium anomalies if they were present. They're not present in this case, regardless of the method used. The paper I mentioned recently, Zhang and Shields, came out this year in Chemical Geology. They've shown, they've taken a different view and suggested that actually rare earth elements are quite susceptible to diagenesis, and that exchange is possible even in some cases where other proxies like carbon isotopes are well preserved. And they've suggested that anoxic pore waters and aragonite are more susceptible to this, which means, of course, that there could be some systematic bias over Earth history in bits of the rock record that either are or are not recording primary seawater signals. They suggest, and I completely agree, that a really good way around this is to take account of the entire pattern and to cross-plot cerium against all of the other rare earth element parameters and the concentration of other elements like aluminium and zirconium. So this is something that I would strongly encourage anyone who's working with cerium anomalies to do. You have to present somewhere the entire rare earth element pattern, and you have to consider whether your negative cerium anomalies or positive cerium anomalies, if they're present, correlate with any other parameter like the lanthanum anomalies or the yttrium anomalies, or the total concentration of rare earths or the europium anomalies. There's actually a whole number of different parameters that you can look at within the rare earth element pattern. And often, in the main section of a paper, what I see presented is, for example, the cerium anomaly versus stratigraphic height. And sometimes you see a nice trend, for example, like an increasing negative cerium anomaly with time. And then when you go and dig into the supplementary information and you look at the rare earth element patterns, you see that the negative cerium anomalies occur in the samples with the lowest concentrations and the otherwise best preserved rare earth element patterns. And that where there's no cerium anomaly present, you've got more of a clay signal. And so actually what you might be looking at is that rather than a systematic change in primary redox conditions, you had a systematic change in lithology and an increase in clay content, and so increasing clay contamination of your signal with stratigraphic height. So analysis, so there's the digestion technique that you have to do in the lab, which is outlined in a number of papers. You just add, it just involves pipetting and centrifuging and centrifuge tubes. It's pretty straightforward and low cost. Then you analyze your solutions using ICP-MS, or you can use laser ablation. There's some issues with laser ablation with getting your rare earths above the detection limit. You have to use a pretty big spot size, and sometimes even then it's not always possible to get good data. Then you do the data corrections as you would with any ICP data, except you have to take account of the sequential digestion method that you used. So there's just an extra little correction to do there, but that's all just pretty straightforward maths. Then you have to normalize to shale, PAAS. And then finally, as well as plotting the patterns, you have to start calculating some anomalies. So here's some common ones, but there's actually loads out there. You can get really carried away with doing ratios of all kinds of things. But the most important are the sum of the rare earth element concentrations. So this is the sum of the shale normalized concentrations, but it excludes yttrium. The yttrium to holmium ratio, and this uses the non-normalized concentrations, or at least if you're not doing that, you have to specify whether your concentrations were normalized to shale or not. And something quite confusing in the literature is that sometimes people use mass ratios and sometimes molar, so you get slightly different numbers. So just be careful to report that and to think about that when you're reading papers. The ytterbium to neodymium ratio is a measure of the heavy rare earth element to light rare earth enrichment. And the bell-shaped index, BSI, is an indicator of to what extent your rare earth element pattern has a bell shape like this. So it's the concentration of the middle rare earths divided by the light and the heavy. And this, of course, you have to exclude the elements that are independently anomalous, such as cerium, europium, and yttrium. And this can indicate contamination from phosphate minerals. Also with these anomalies, there's some different formulas out there. So in the old literature, sometimes they use these formulas where you calculate cerium by simply going 2 times cerium divided by the sum of its neighbors on either side. And the same for europium, where you just divide it by the sum of its neighbors, samarium and gadolinium. But unfortunately, both lanthanum and gadolinium can also show anomalous behavior, right? And what we're trying to do is quantify the concentration of cerium relative to the hypothetical concentration if there wasn't any redox-related cycling. And that requires you to look at the trajectory of the non-anomalous rare earths when they form this nice, smooth slope, and project where cerium would be along that line if it wasn't behaving in an anomalous fashion. And so a better way to do this is to use these revised formulas from Lawrence. I should have put the reference, sorry, I think it's Lawrence 2006, which is cerium divided by praseodymium squared, which is divided by neodymium, and a similar story for europium. So you're just normalizing to the non-anomalous neighbors on one side instead. Another thing you commonly see in the literature is a cross plot of cerium anomalies versus praseodymium anomalies. And this was something people did when they were using this old formula so that they could distinguish true cerium anomalies from those generated by just an enrichment in lanthanum. So if you're using the new formula for cerium anomalies, it's not necessary to do those plots. You'll basically get a straight line because the praseodymium anomaly is just an inverted version of this. But yeah, people often still do that. It might be useful to just so that everybody's got a good comparison to calculate it using the old formula and cross plot it with praseodymium anomalies and calculate it using the new formula as well. So I'll just go through a couple of case studies now. The first one is from our work in Namibia, which is from a while ago from my PhD. So the Nama group is a carbonate ramp system deposited in the Ediacaran, which hosts early animal fossils and the first skeletal fossils. And this is our field site in South Africa. This is the Orange River here, which borders Namibia and South Africa. So the other side of the river is Namibia over there. And my master's student, McDonald, who's now graduated, who was looking at strontium isotopes. So this is just a little case study of how we can apply this in deep time. So we analyzed rare earth elements using a sequential digestion technique from 9 different sites across the basin. And these are the cerium anomaly data in black and purple, and they've been culled so that I'm only using patterns where there's convincing evidence for preservation of a primary seawater signal based on all of the other anomalies and parameters that I mentioned. We also did ion speciation in carbonates, which is sensitive to the onset of ferruginous anoxia, and we combined the 2 proxies to try and distinguish between fully anoxic, intermediate manganese, and fully well-oxygenated conditions. So this is the same 9 sections, but with those data interpreted in each case and plotted against the distribution of fossils. And what we found when we did this, which I think is also quite convincing that the proxies are telling us something about the primary redox conditions. When you look at the distribution of redox conditions, they correlate beautifully with the presence of fossils. So where we have sort of mixed redox conditions in a ramp environment with rapid oscillations backwards and forwards between anoxic, intermediate, and oxic conditions, we find that where we do have thin horizons of fossils, they correlate. Sorry, I didn't catch. Was that a question for me?
I think not. I think that was just unmuted.
Okay, fine. Yeah, where you have a short-lived oxic horizon, we find that's where the fossils are. And then where we have more long-lived, persistent, well-oxygenated conditions, we start to see things like larger body sizes, a mixture of soft-bodied and skeletal biota, and reef-building behavior. We can also look at long time scale compilations of cerium anomalies in carbonate rocks. And even though the signal is telling us very much about local conditions on the shelf, if you compile enough data over geological time, you might start to see a significant pattern emerging that's telling you something about global availability of oxygen. And so this is a compilation based on Wallace and Hurd and Tang, showing that there isn't a significant change in the frequency of cerium anomalies until well into the Phanerozoic. And that largely throughout the Precambrian, we don't have any significant negative cerium anomalies. And in some cases, we have positive cerium anomalies. So a point here about positive cerium anomalies, they often get slightly misinterpreted. So if you have a positive cerium, it's telling you that that sample locally must have been deposited below the chemocline. So you had the dissolution of manganese oxides and buildup of excess cerium, but it also tells you that there has to be oxygen somewhere above you, right? So in a water column with absolutely no oxygen available, like perhaps we had in the Archean, you wouldn't see any negative or positive cerium anomalies. Everything would be 1. If you have a stratified water column with oxic conditions above and anoxic conditions below, you can have a negative cerium anomaly above and then a positive cerium anomaly below the chemocline. So just to make that distinction between what it's telling you about the local conditions and what it's telling you more broadly about the regional and global conditions. Although maybe when looking at this plot, we should bear in mind the point that Webb made back in 1999, that actually the partition coefficient for cerium is slightly greater than the rest. So maybe an average of 1.1 is telling us that seawater had an average of 1. So this cerium compilation showing no significant change until the Paleozoic fits in with a number of other compilations of local redox proxies like iron speciation, iodine over calcium, which also show no statistically significant difference until the Paleozoic and with COPS biogeochemical models. The global proxies tell a far more complicated story of oscillating redox conditions across the Neoproterozoic and Paleozoic. But I think that, you know, these data themselves, because they make sense and they fit with other redox proxies and they fit with the distribution of biota, that it's kind of convincing evidence as well that there is a primary story being told by Cerium in the rock record. That does not mean that every single Cerium anomaly is primary. There's definitely cases where you've got false positive and false negative Cerium anomalies due to the local conditions. But I think on the whole, I still think that in carbonates, this represents a very promising proxy for extracting primary signals from the oldest parts of the rock record. I'm going to finish with another case study on something quite different. That's current work by my PhD student, Anselm K. He's been working on BIFs from the Transvaal supergroup, which were deposited in the final stages of the Archean. This is a photo from our field site where you have some of the most beautiful carbonate rocks and microbial textures I've ever seen on the lower half of the copy, and then altered BIFs at the top. But the samples in this study are from core. So they've not been altered and oxidized to the same extent as this red BIF we're seeing here in the field. And the core samples that we've been looking at preserve a lot of very fine iron silicate minerals, including greenolite, and we think that these were primary, in line with a lot of Berger-Rasmussen's interpretations. And there's recently a couple of models that have emerged for how these iron silicates were being precipitated. So if we accept that they were, at least in some cases, a primary orthogenic mineral and that they were an abundant component of seawater, you know, perhaps not to the exclusion of other important primary minerals in seawater. There's a whole big debate there, but I think greenolite was certainly an important primary component of seawater in the Archean, component of the sediment record. The next question is, how are these needles forming? And one suggestion that's come about recently from modeling work by Tutelo and Tosca, and also some higher temperature experimental work, which is a paper of mine in Press, suggests that they could have formed very rapidly at hydrothermal vents and then been transported onto the shelf by hydrothermal plumes or just lateral transport. And other models suggest that they precipitated directly on the shelf, so rained down from above from seawater, perhaps driven by small amounts of iron-3 in seawater or transformation of green rust, or small changes in pH. So there's a number of models that have been put forward, but we could broadly divide them into hydrothermal vent models and shelf models. So my student has found what he thinks is really nice evidence of primary greenolite needles and analyzed them using laser ablation. And the first thing you'll notice, so these are the patterns here in green. This is an average. The black line represents modern hydrothermal fluids and the blue line represents modern seawater. And he's comparing also to a number of studies from the literature where they've got bulk data from iron formation. And actually, if we look at this plot, it's probably more useful, the total rare earth elements versus the europium anomaly, which are the two major parameters that differ between hydrothermal fluids and seawater. You can see hydrothermal fluids have way higher rare earth element concentrations and way higher europium anomalies. These are both log scales. And our data are clustering in line with bulk BIF, in line with carbonates from the time, and much closer to seawater. So what we would expect in the Archean Ocean when hydrothermal fluids were, the fluxes were higher and they contained higher iron concentrations, slightly different chemistry. We'd expect that europium anomaly, which is characteristic for hydrothermal fluids, would start to mix and persist in seawater and be present everywhere at low levels, right? So you'd expect to see a small europium anomaly of, for example, 1.5 all throughout Archean seawater. what we're not seeing is evidence that greenolite was forming directly from a hot hydrothermal fluid, which has europium anomalies of like 36. So I think this is perhaps evidence is still very preliminary. He plotted this data in the last week, so we're still thinking about this. But I think this is another nice example of how you can use rare earths, not just the cerium anomaly, but the whole pattern, the concentrations, and the europium anomaly, to ask other questions like the provenance of important minerals. Yeah, so with that, I'll just thank my group and funders, and very happy to take any questions.
That was great.